Good morning, it's June 9th, and this is your daily brief in science. Here's everything you need to know. Bowel cancer has emerged as the second leading cause of cancer deaths in the UK, accounting for approximately 16,800 fatalities annually, with 1,700 of those occurring in Scotland. Alarmingly, the disease is increasingly being diagnosed in younger individuals, prompting urgent attention from the medical community. Recent studies published in The Lancet Oncology indicate that early-onset bowel cancer rates are rising in 27 out of 50 countries, with a notable surge among young women in Scotland and England. Research has revealed that bowel cancer cells are adapting to resemble more resilient skin and muscle cells, which enhances their survival in harsher conditions and facilitates their spread throughout the body. A significant factor in this process is the ATRX gene. Its loss has been linked to an increase in metastatic tumors that spread to critical organs, such as the liver and lymph nodes. These findings, published in the journal Nature, highlight the complexities of bowel cancer metastasis and the need for innovative treatment strategies. The research aims to inform the development of new treatments that could prevent this cellular transformation, which is crucial for improving patient outcomes. Dr. Catherine Elliott, Director of Cancer Research UK, emphasized the importance of early diagnosis and treatment, advocating for ongoing research to discover methods that can prevent cancer spread. In response to the rising rates of bowel cancer, Cancer Research UK has invested £5.5 million into initiatives like CRC, STARS, which brings together over 40 experts to find innovative and less harmful ways to combat the disease. Dr. Kevin Meant stressed the necessity of understanding the disease's growth, especially given the increasing diagnoses among younger individuals. Furthermore, Dr. Patrizia Camerari noted the implications for treatment strategies, as cellular plasticity plays a crucial role in bowel cancer metastasis. Targeting the shape-shifting ability of cancer cells could lead to new treatments that effectively prevent cancer spread, addressing the urgent need for effective therapies in light of the approximately 4,000 new bowel cancer diagnoses each year in Scotland alone. Parkinson's disease is a progressive neurological condition that affects millions, characterized by symptoms including tremors, depression, balance issues, sleep disturbances, and memory problems. Alarmingly, about 1/3 of Parkinson's patients may eventually develop dementia, raising concerns about the overall health management of these individuals. As the disease progresses, many patients become increasingly reliant on caregivers, which can lead to neglect of basic health practices such as oral hygiene and nutrition. This highlights the critical need for proactive management to prevent further deterioration of their health. Dr. Saeed Shoai has emphasized the important connection between gut and oral bacteria and neurodegenerative diseases. He noted that disruptions in the gut-brain axis can lead to inflammation and neuronal damage, making the maintenance of oral and gut health a potential strategy to mitigate neurodegenerative processes in Parkinson's patients. A recent study published in the journal Gut Microbes analyzed 228 samples from various patient groups, including those with mild cognitive impairment and dementia. The findings revealed that toxins produced by harmful bacteria could serve as markers for identifying Parkinson's patients at a higher risk of developing dementia. Dr. Frederick Clayson pointed out that it remains unclear whether these bacteria cause cognitive decline or if changes from Parkinson's enable their growth. However, he suggested that these harmful bacteria may contribute to the worsening of symptoms. The study's results could pave the way for targeted treatments aimed at altering the gut environment to protect brain health. Promoting a healthy microbiome through oral care, diet, and probiotics may improve disease management and overall health outcomes for Parkinson's patients. Researchers are also utilizing artificial intelligence to connect shifts in bacterial populations to cognitive decline in Parkinson's patients. This innovative approach has identified specific bacterial species that were previously undetectable through traditional testing methods, offering new insights into the disease. A recent study has upended long-held beliefs about the Tonto Group rocks in the Grand Canyon, offering new insights into Earth's geological history. Published on June 7, 2025, This groundbreaking research reveals significant shifts in fossil assemblages within the Tonto group, showcasing distinct trilobite communities that indicate rapid evolutionary changes across sedimentary layers. Researchers discovered evidence of multiple shoreline advances during the Cambrian period, challenging the notion of a single rise in sea level. Instead, the Tonto group is now understood to have formed through episodic surges of seawater and rapidly changing environments. These sedimentary layers are critical for understanding ancient oceans and ecosystems that have since vanished. Carl Karlstrom from the University of New Mexico notes that the new model presents a mix of marine and non-marine environments, along with faster evolutionary changes than previously believed. James Hagedorn from the Denver Museum of Nature and Science adds that this research enhances our understanding of the Cambrian explosion and influences global interpretations of rock records. Using uranium-lead dating on zircon crystals, the team correlated rock layers and found that significant faunal changes occurred in less than 800,000 years, a much shorter time frame than earlier estimates. The study emphasizes the importance of understanding geological processes in light of modern climate challenges, as they provide insights into how past environments responded to change. This reinterpretation of the Tonto group highlights the provisional nature of geological truths and the need to integrate fossil records, sedimentology, and high-precision dating techniques. Key findings suggest that the Cambrian period was marked by swift ecological shifts, altering our understanding of early life on Earth. The study was published in GSA Today, showcasing the value of modern scientific techniques in reinterpreting long-standing geological formations. China's ambitious space exploration continues with the launch of the Tianwen-2 spacecraft, which took off on May 28, 2025. This mission is focused on exploring the near-Earth asteroid Kamo-Walewa, a celestial body of significant scientific interest. As of June 6, 2025, the spacecraft is over 3 million kilometers from Earth and is reported to be operating well by the China National Space Administration. Tianwen-2 is expected to reach Kamo-Walewa by July 2026, where it will spend several months orbiting the asteroid to gather crucial data. Kamo-Walewa is classified as one of Earth's 7 known quasi-moons and is believed to be a fragment of the Moon, making it a unique target for study. The spacecraft has already transmitted its first image back to Earth, capturing one of its solar panels, which resemble those used in NASA's Lucy mission. Equipped with a suite of scientific instruments, Tianwen-2 will analyze the asteroid's composition, shape, and other characteristics. The mission aims to enhance our understanding of the solar system's evolution and the origins of water on Earth. This endeavor marks China's second planetary exploration effort, following the successful Tianwen-1 mission to Mars in 2020. Tianwen-2 is part of a series of successful missions by China's space program, showcasing its growing capabilities in deep space exploration. After completing its sample return mission, the spacecraft will utilize Earth's gravity to slingshot toward Comet 311P/Pan-STARRS with an expected arrival around 2035. The data collected from this mission will contribute to global scientific knowledge and solidify China's position as a leading player in space exploration. A recent study from the Earth Life Science Institute in Tokyo, in collaboration with the Hebrew University of Jerusalem and the Weizmann Institute of Science, has unveiled a remarkable discovery in molecular biology. Researchers investigated an ancient protein known as the helix-hairpin helix, or HHH motif, which exhibits a unique property termed functional ambidexterity. This means that the HHH motif can bind to both standard and mirror image versions of DNA and RNA, a finding that challenges established norms in the field. The study revealed that the mirror image HHH motif successfully binds to double-stranded DNA, contradicting the expectation that its reversed-handedness would impair its functionality. This is significant, as life on Earth typically demonstrates homochirality, where proteins are made of left-handed amino acids and nucleic acids consist of right-handed sugars. The research team initially faced skepticism regarding their hypothesis, which they ultimately validated through laboratory experiments and molecular simulations. These experiments showed that the binding kinetics and mutation effects were surprisingly similar between the natural and mirror image proteins, suggesting a potential link in their binding modes at the molecular level. The researchers speculate that the HHH motif's ability to function in both handed forms may provide insights into ancient biological processes, possibly indicating the existence of mirror-image life forms in the distant past. This study is the first to document ambidextrous proteins binding to nucleic acids, prompting further investigation into the evolutionary implications of this phenomenon. The findings challenge long-held beliefs about molecular design, suggesting that this nucleic acid-binding protein can operate in what could be termed a mirror world. Led by Professor Liam M. Longo and Professor Norman Matanis, the research was published in the journal Adjuvanta Chemie and opens new avenues for exploring other proteins that may share similar ambidextrous characteristics. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.